Welcome to the eukaryotic cell cycle and mitotic cell division. Before we jump in, let's look at how this relates to humans. When would humans do this mitotic cell division stuff? And one time we do this is way back in history. If you remember, we talked about fertilization and how it produced this diploid zygote, which is the first cell that we ever were. And that zygote reproduced through mitotic cell division to make more and more and more and more cells. And this is where mitotic cell division comes into play. Each of those cells were identical to each other. They had the exact same DNA. And eventually they went through the process of differentiation. They turned on and off different genes to create all the different types of cells and tissues inside of your body. There are other times that we use mitotic cell division, and that involves the replacement of cells. So for example, if you're losing skin cells, or you get a cut, or you damage the lining of your stomach, for example, you're always making new cells through mitotic cell division. There are unicellular organisms who reproduce this way through mitosis to make copies of themselves, and every single time mitosis happens, you end up with two genetically identical cells. It's a form of asexual reproduction. Just as we have a life cycle, there is a cell life cycle known as the cell cycle. And it consists of mainly two stages. There's interphase and there's the mitotic phase. The cell spends most of its life in interphase. Here it's really growing, it's increasing its cytoplasm, it's getting bigger, it's making copies of its DNA, and it's just waiting. This is where the cell spends most of its life, especially a healthy cell. Cells that have turned cancerous actually don't spend enough time in interphase. In the second stage of the cell's life, we have the mitotic phase. And during this phase, there's actually two parts. So the cell is dividing during the mitotic phase, and that dividing happens through mitosis. And mitosis is the division of the nucleus, the brain of the cell. And since we don't want to end up with one cell with two nuclei, we have to divide everything else, and that's where cytokinesis comes into play. So here's a nice, useful graphic showing you this is most of the cell's life, this is interphase, and here is the division phase, the mitotic phase, that involves both mitosis and cytokinesis. Okay, now interphase looks like this on a microscope, and some things that you can see about interphase is the nucleolus. You can see the nucleolus, those dark spots. You can't see chromosomes yet. The DNA is all jumbled together. And we're going to take a look at this picture here. You can see the jumbled together DNA, and you can see the nucleolus. Now, interphase comes in three steps. There's the G1 phase, and during this part of the cell cycle, the cell is just growing and it's increasing its cytoplasm, it's getting bigger. During the S phase, this is the DNA synthesis phase, the cell is actually copying all of its DNA, because if it's going to divide, the two new cells are each going to have their own copies of the DNA. And then during the G2 phase, the cell is going to grow some more and get ready for cell division. So that's interphase. After interphase happens, we can go on to the mitotic phase. We're going to look at mitosis. It turns out that that mitosis is not just one step. It's actually a series of steps, beginning with prophase, proceeding to metaphase, anaphase, and then telophase. And we'll look at each of these one at a time. And then again, as soon as mitosis is done, and mitosis is the division of the nucleus, cytokinesis is going to happen. And cytokinesis actually overlaps with telophase. They kind of occur at the same time, and we'll take a look at that a little bit closer. So before we jump into mitosis, we have to know some terms, and one of those is the spindle. This is the mitotic spindle. It's going to be really important for pulling apart sister chromatids from each other, which we'll look at next. And there are these things known as the centrosomes. The centrosomes actually make the spindle, and the centrosomes in animal cells have centrioles, which you learned about as one of the organelles. And centrioles are kind of mysterious. We don't know exactly what they do, but we think they help in the formation of cilia and flagella in cells that have them. Okay, so this is just a little bit of vocab. So we finished interphase and we're ready to jump into mitosis. The first step of mitosis is prophase. So we did this, we did interphase, and this is prophase. So during prophase, the chromosomes become visible. So you can actually see those dark chromosomes now because the chromatin has condensed. And just as a reminder, this is what a chromosome looks like. it's made up of two sister chromatids, right, which are copies of each other. And that copying happened during the S phase of interphase. And the sister chromatids are held together by the centromere. And you can see those X-shaped structures over here on the sketch. Okay, other things that happen is the nuclear membrane starts to dissolve and disappear. The nucleoli disappear. You can't see those dark spots anymore. And the centrosomes move to opposite poles. And the spindle microtubules are going to start binding to the centromere of the chromosomes. After prophase, we enter metaphase. And during metaphase, the chromosomes line up in the center of the cell along the equator, along the metaphase plate as it's known, and they're in a straight line with the centromeres in the middle. You can see that over here, metaphase taking place, and you can kind of see the spindle fibers here too. After metaphase happens, we enter anaphase, and during anaphase, the chromosomes are going to be, the chromatids, the sister chromatids are going to be ripped apart. They're going to be pulled apart, and they're going to move towards opposite poles of the cell. And this is happening because of the spindle fibers helping to pull them. The cell is also going to elongate, and you can see that happening here. Anaphase, you can see the sister chromatids being ripped apart and being pulled towards opposite poles. After anaphase, we enter telophase. The cell is continuing to elongate, and a nuclear envelope is forming around the chromosomes at each pole. So you have a new nuclear envelope forming. You have the nucleoli starting to appear. You have the spindle going away, starting to disappear. So here you can see telophase happening. You can see the new nuclei forming here. And also those chromosomes are going to start to become loose again instead of condensed. So we divided the nucleus, right? We did all those steps. And now we are stuck with two nuclei in one cell, which we don't want. So we're going to engage in cytokinesis. And in this part, you're going to have the cell kind of pinch inwards and divide the cytoplasm so that you have two separate daughter cells, and they're called daughter cells. So we just did this, prophase, metaphase, anaphase, telophase, where we divided the nucleus, and now we're dividing the cytoplasm. And if you look at the photo that we had, telophase and cytokinesis kind of happen at the same time, and you can see the cell wall forming right over here because cytokinesis and telophase are kind of overlapping. So there are some differences in cytokinesis between plant and animal cells. In animal cells, cytokinesis involves something known as cleavage. So a cleavage furrow is going to form from a contracting ring of microfilaments, and it's basically going to pinch the cell. So that furrow is going to deepen and deepen and deepen, and it's going to separate out the two cells until you end up with two daughter cells. So this is how it takes place in animal cells. Plant cells, on the other hand, and we have been looking at photos of plant cells all along, what happens here is a cell plate forms in the middle, and the cell plate forms from vesicles containing cell wall material. You can see the cell plate forming right over here, and it's going to grow outward towards the edges, and it's going to divide the content of the cell so that you end up with two daughter cells that are next to each other, just like this. So let's take a look at mitosis in action. This is an animal cell, and you can see a time lapse of mitosis, if you will. So it's pretty cool. You can see the chromosomes, and they are getting organized. All right, we are trying to enter metaphase here. We're trying to line up in the center of the cell. There it is. Looks pretty good. It's important that the cell gets this just right so it can evenly divide. And now you see the anaphase taking place. You see the sister chromatids being ripped apart, and now you can see the cell is being split into two separate daughter cells. So that was pretty cool. And the chromosomes are going to become loose again. So I hope that was interesting and helpful. All right, in summary, this is what we've done today. We looked at the cell cycle. We saw that the cell spends most of its life in interphase. This is G1 phase of interphase. The cell is growing. Here the DNA is making copies. And here the cell is growing and getting ready for cell division. Here, in the purple and the pink, the cell is actually dividing, and dividing involves mitosis and cytokinesis. And we know mitosis comes in 4 steps: prophase, metaphase, anaphase, and telophase. So I really hope that was helpful. Thank you for watching.